hello everyone. So our project was training a neural net to identify if there are point mutations that conferred significant dependencies in cancer cell lines. So let's start with the definition, and that's what a dependency is. So there are genes in the genome that are essential for cell survival and function. So if you knock out said genes, then the cell dies, and you say the cell is dependent on that gene. That could be a good avenue for therapeutics in cancer biology, because if you target a gene that's necessary for a tumor to survive, boom, you've killed the tumor. However, a lot of genes that are essential for tumors may also be essential for normal cells as well, meaning that this could be a highly cytotoxic approach. So one insight is that identification of point mutations in tumors is at the exclusion of their presence in normal cell lines. So if we can identify point mutations that have significant dependencies in contrast to knockouts in the same gene lacking such point mutations, we can identify precise targets for therapeutic intervention utilizing CRISPR.
Using this insight, we identify significant different dependencies in cell lines with more than 5 mutations and more than 5 wild type cell lines to construct our training data. And then we train our neural network model to predict the genes that have mutations that significantly impact dependencies on those genes that have fewer than 5 mutants and wild type cell lines.
So our first pass was first to identify if these significant dependencies existed at all. So you take your 5 mutants, your 5 wild types at minimum, and then the dependency scores distribute as a normal distribution, so you can simply define with an FDR-corrected p-value less than 0.1 which genes had significant dependencies in their point mutations. And we found about 34 of these genes do in a tissue-specific context. And if you notice, there's a lot of overlap across tissues for certain genes. And in fact, if we look across all tissues rather than restricting tissue by tissue, 6 genes come out as also significantly dependent in both contexts. All of these genes are found in common oncogenic signaling pathways, such as RAS and PIK3CA, which is really exciting because what this suggests is addiction to survival and proliferation pathways. All of these mutations are not uninducible, suggesting that knocking these out reduces survival and proliferation of cancer cells and would then stem metastasis. So on to our training.
Yeah, so we train our data using several machine learning technique approaches, such as random forest, as SVM and neural net. And we found that for all this machine learning algorithms, they were very accurate and specific. However, they were not so sensitive, unlike the neural net that was more sensitive to unlike the other ones. We also look at the importance of our features, and we found that the mean expression, the proportion of TCGA hotspots were more important, which actually agree with some with the literature that these features were more predicted than any other ones.
These are our predictions.
Yes, so using our best model, which is the neural network, we identified that the majority of the predicted significant mutation dependencies for each gene, they were mutually exclusive, meaning that they were in one tissue compared if they were in multiple tissues.
Yeah, which then suggests that we're getting to our long tail of discovery and identifying rarer events, which is also quite useful. Now, the thing is that cell lines are highly heterogeneous, and there's no guarantee that what you observe in cell lines can even be found in tumors. So we look to TCGA in the specific tissues in which we found significant dependencies to see if our predicted dependencies were actually detectable, and 33 of them were. And we actually decided to go a little further beyond to see if any of these point mutations on their own impacted the survival of these TCGA patients. And in glioma, we had one such example, which was PIK3R1, which is the single most recurrent and mutated gene in glioma, as well as the fact that all of these mutations are loss of function mutations in glioma, which suggests that since we know if there were also a point mutation in these samples, our data suggests and our results suggest that in fact the prognosis may further improve for these individuals. So what have we learned today? Well, what we've learned is that we're able to train a pretty specific and somewhat sensitive neural net in order to identify significant dependencies. The PIK3R1 mutation actually was predicted by our model rather than in our earlier pass, but that earlier pass was informative too in identifying that cancer cell lines can be addicted to oncogenic signaling mutations. And furthermore, since we only have 50% sensitivity, this means that the long tail still eludes us just a little bit. So we want to increase our sensitivity further. Something that we did but didn't show the results of is that we tried to integrate the CCLE epigenomic information, the bisulfite sequencing, and the proximity to promoters. That ended up not really increasing the margins of our sensitivity, or was very predictive according to our random forest model. But this suggests that maybe we need to look outside of CCLE for information on cancer cell lines. So ENCODE could be a good resource for getting such features. And so that we're happy to take any questions. Josh, you have a question?
Yes. So presumably you don't actually need that to identify that many good mutation candidates for CRISPR to work. So how important is it actually to increase the sensitivity?
Well, it's a good question, and I think it's to further understand that there are instances, sort of what we heard about earlier with this cancer of unknown primary. There's also this idea that clinicians will be able to diagnose tumors with some form of driver mutations, but those driver mutations may not actually be the targetable CRISPRable mutation. So the idea is that maybe it's something that's actually extremely rare in the population, but incredibly powerful.
Do you have any information about the mutations that were there when the cell line was first started? I'm curious about the original tumor versus all of the additional mutations that have made these cell lines a success.
So we talked about this in our final report, how this is one of the limitations of our work, is that the timing is not easy to resolve because none of these are longitudinal samples. But what would be a really great future direction, and one that's somewhat close to my heart, is if you were to take longitudinal samples and if you were to then look at the timing of when such mutations occurred in clonal evolution, if the order was important.
But also you could resequence all the existing lines.
Yeah.
See how does that compare to the sequencing experiment from 2 years ago?
Yeah, and that would improve the KM.
That would get at the sort of temporal aspect of someone.
Yep.
Chris, thank you. So next, are you guys leaving early?
Yes.
Yes, you are.
Yeah. Team 21. Please pull it up.
Oh, yeah.
Yes.
Do you guys like cavemen?
Any minute?
Well, it looks like a cave. Yeah.
Get a roll cake with the very wrong. I think now the
Yeah, this is fine. So